Well, it's a great pleasure to take part in this. I had already, even without Jane Litzio being so heavily involved, I had heard of this group and know how important it is for anyone coming, particularly those with families. So this is the title, and I want to just reassure you before you all hasten to leave, which is what many scientists do when they hear there's an immunology talk, that it's going to be very, very illustrative. And it's basically a series of movies and pictures by which I want to tell a story, which tells you something about the science that underpins the way our bodies fight cancer and also COVID-19. So here is a really beautiful picture taken by one of my students of this very spiky killer cell, which is in our immune system, and it's recognizing this target cell here, and you can see it latches on. And what I'm going to tell you the story about is how does this cell pick out and kill right targets, and how does it help defend our body? But I had a suspicion that I would be given a rather illustrious introduction by you. And so I thought I had better take you back to my start about why I became a scientist. So here I am, age 6, with a beautiful present given to me by my aunt. And I am here, this was like the best present I ever got. And you can see that I have this inclination towards medicine, and I'm here bandaging my mother's arm, and my brother is waiting in the queue. My parents were both school teachers. My mother was a primary school teacher. My father taught English. There was really no scientific pull within the family, although perhaps I was influenced by this how glamorous the nurses' uniforms looked in those days. But of course, education was highly valued in a family full of teachers. But I think one of the reasons that I became a scientist and not an English literature person was because I was lucky to hit a phase within the UK where there was a rather special biology and in fact chemistry O-level syllabus, as GCSEs were called in those days. And it was based on learning from experiments. And nowadays everything's online, and so it's possible to go online and find your old textbooks. And I was delighted to find the Nuffield biology textbook. And I wanted just to tell you, to show you a couple of little excerpts from it that were really underpinned why it captured my imagination. So the first is that they say, although doing an experiment is important, nothing can be discovered unless the scientists watch what happens. So here's an example. Now, try answering these two questions without looking around the room you're in. How many ceiling lights are there in the room? What is the color of the door handle? And I mean, you may be able to answer these if you're in your own home, but it's not easy to notice something. And this can happen when you're watching experiments just as easily as it can happen at any other time. So this, together with the teachings of Cesar Milstein, who was my PhD advisor, who always taught me to look at the details, really, I think has been one of the largest influences on not just my fascination with science, but the way I do science by really looking at the details. You can easily see things that most people have missed, and it's the details that are the most informative, in my opinion. So, having set the stage, this is my excuse for showing you really what will amount to a series of movies and images to explain to you the science that underpins our immune response to cancers and viruses such as COVID. So it's been a horrible year. It's been horrible for many of us in many different reasons, but it is possible to find some positive aspects of the pandemic. And the positive aspect for immunologists is that everybody learns in immunology. You can open more or less any newspaper and find that people are talking about antibodies and T cells. And you know, people from all age groups, from children, small children up to sort of older people. And I just wanted to remind you, just in case you missed this or you hadn't found something that explained it simply enough, because actually when I trawled through Google trying to find a nice image that would illustrate how much has been taught through the COVID pandemic, I couldn't actually find a very nice, simple diagram. They're all, in some ways, rather complicated. So here is my very, very simple diagram about all the immunology you need to know to follow the rest of the talk. And that is that when, for example, you have a virus-infected cell, so here are my little viruses that I drew, and were the nearest thing I could draw to coronavirus, but I'm not a very talented artist, as you'll learn. So here is your virus-infected cell, and the virus has bound to a receptor on the outside of the cell, and it's internalized, and now it's multiplying like crazy inside the cell, and this is the cause of the illness. So it then secretes the viruses out again, and then they can go off and infect more cells. So that's what you need to try and stop. So there are two main types of cells that do this. There are B cells, which is what I worked on when I did my PhD with Cesar, and the B cells make antibodies, and they're proteins that can be secreted, and they recognize something specific about a cell or a virus. And so here you'll get some B cells that will make antibodies to the coronavirus, let's say. And so that cell will then be stimulated, saying, oh, there's something out there to recognize, let's make more B cells, let's make more antibodies. And that's how you get antibodies. And then the antibodies can bind the virus, and they can block it, because now when it's all coated with antibody, it can't bind its receptor and infect more cells. So that's the simple way antibodies work. Then, of course, the focus of my talk is going to be the killer cells. And killer cells come in and they recognize a virally infected cell, usually by a little bit of the virus that is appearing on the cell surface. And they can come in and destroy that virus infected cell. And I think my animation might go so far to see this is my destroyed cell. So it's been killed. You kill the whole cell, but you get rid of the virus that way. So really the story is very, very similar in cancer, that cancer cells look a bit different. There's something not quite right about them. And there are changes that occur when the cells become cancerous. And these can be detected by T cells. And once again, what happens then is that the killer cell can come in, you can see that looks different, that cell, and again, it destroys that cell. So it's the same cell that's being a superhero of both virus infections and cancer. So the technical terms, which we've already heard. So in technical terms, and you'll see this on some of my slides because I've just imported them from my scientific talks, that these killer cells are known as CD8 positive cells because they have a marker on them called CD8, or cytotoxic T lymphocytes, which is often abbreviated to CTLs. So you'll see these terms on some slides, but I'm basically talking about the same killer cells. So what I want to tell you today is, what do they look like? How do they work? And why is it that they're revolutionizing cancer treatment? And in fact, are they working with COVID-19? So here we go. This is the 5 million potential killers in a teaspoon of blood. And you can see they're relatively small cells. They're very motile. They move around. And they can patrol your body, seeking out cells that look different and then destroying them. They're incredibly good at what they do. They can not only recognize one cell, but having killed it, they can go off and kill others. They are serial killers. So what I want to do is to try and zoom in a bit on how they work. So in this slide, so the movie again, you can see in red these are all cancer cells that have been put in a dish, in fact, that we can image with our microscope here. The green cells are the killer cells, and there's a little red dot inside them that shows you something inside that I'm going to tell you a bit more about, which is involved in the killing. So here, if I set the movie going, you'll see we're going to zoom in on this killer cell here that has recognized the red cancerous target. And you'll see it probes, it latches on, it probes the surface. And in fact, you can see many others latch onto this target too, and they all come in and they very rapidly, in a matter of minutes, destroy this cancerous cell. So what does it look like? If we capture a still moment, we can see what's going on. So here's 3 different killer cells latched on to a single cancer cell, as you saw in the movie. In this case, the cancer cell happens to have been dyed green, and this is its big blue nucleus. So they make a very, very, very organized interface with the cancer cell, and they organize all the little receptors that can recognize that cancer cells and all the little receptors that can latch onto that cancer cell, and they put them in a very specific, if you like, suction couple, more or less, that allows them to say, yes, this is right, we're going to go ahead and kill. And then, this interface has become known in the immunological community as the immunological synapse. You can see that occasionally in my slides. But what you'll also notice are that there are these little red, rather unusual secretory granules within these cells. And these secretory granules are often known as cytolytic granules, because cyto means a cell, lytic means you can kill it or lyse it. So when these granules release their contents, then they can kill the cancer cells, and that's what really makes these cells so powerful. So what happens? I'm just going to show you a cartoon to really show you the moleculars we're going to get, why these secretory granules, again, really captured my interest when I was starting out my own research lab. So these really are not really meant to be secretory granules. They're meant to be a compartment in the cell, which is known as the lysosome, which I'm sure Jane can fill you in on more because her husband works on lysosomes. But lysosomes are, in effect, the recycling centers of all cells. And they are the place where proteins that are no longer needed are sent to be degraded by a series of enzymes that degrade extra proteins that you're just trying to break down so you can take the components and use them again within the cell. But in these killer cells, they've been very sneaky, because not only do they want to package and kill things inside the cell, they want to kill cells that are outside of them, but they use a modification of this same mechanism. So they've added some special proteins which have got the name perforin, which is a name which means a pore-forming protein. And that's exactly what it does. So here's the killer cell in green, and here's one of these secretory lysosomes, and it comes and it forms a seal and releases its contents just at the point in the synapse made with the target it's going to kill. And the perforin can insert across the membrane and then polymerize with itself, making a little donut-shaped pore. And through the donut-shaped pore, it targets another killer cell specific protein, which is the little granzyme, which goes inside, and it finds things it can chop up within the target, and that's the mechanism of the killing. So it's really clever, because it's using a mechanism that all cells use to recycle components by breaking them down and using all the components again, but instead it's added a bit extra so it can get into another cell and kill it straight out. So it not only punches holes in it, but it chops up things inside it immediately. So it's a really effective mechanism of killing. So what does it actually look like? This is what it looks like. So here is our killer cell. Here are our secretory lysosomes in red. Ignore, this is the target cell, which has some red and white coloring, but you can just see the outline of the target cell, long, thin target cell. So what happens as these little secretory lysosome bombs are released is you can see the poor target cell just curls up and dies. This is all a matter of minutes. It's almost eating its way along the cell, it looks like. It's a very long cell in this case. So what you can imagine is very readily is that you don't want these killer cells out on the wild, just randomly recognizing and perhaps killing the wrong cell, an innocent bystander. The killing has to be very precise. So you want these little secretory lysosomes to release their toxins exactly where it's just going to hit the target and not the cell next door. In this case, we haven't got a cell next door. But imagine in the body, cells are packed together very tightly. So it's got to be very precise. So how is it that they manage to aim their killing so accurately? Well, if you take another look at a killer cell killing its target, and this is using an electron microscope where you can see everything inside the killer cell, you can see that it's got lots of special structures within it. So here is the killer cell, and here you can see it poking its way as it recognizes the target cell, which we've colored blue. And you can see, for example, here is one of the secretory granules, which is on its way to the interface where it's going to release its toxins. Here's another one. They look quite different sometimes. And there are all sorts of structures, if anyone has studied biology, that this is an apparatus of Golgi which makes new proteins and reuses the components broken down in the lysosomes. But what I want to focus on is this structure here. In fact, there's two of these little things which are called centrioles. And what these do is in killer cells, they tell the granules where to go. So they say, this is the point where you've got to aim your toxins. And so we know that because we can now zoom out and use a specialized marker that binds to these structures here that we can then image using a video. So here's our specialized marker. So it's known as the centrosome, the two centrioles together. So you can see the two dots of the two centrioles. So here they are, and they usually travel around at the back of the cell. Here's the nucleus, and here's your killer cell, and it's just latched on to this target here, which has a sort of red outside and again a blue nucleus. Just watch what happens to the centrosome. It comes in, rolls around the nucleus, and then it sort of scans. It's looking for the precise point where the toxins need to be aimed. So how does it then focus all of the killing at that one point? So here is here are our two centrioles, the centrosome, and here they are seen in very bright green. And the reason they're seen in bright green is because it's from these little structures, the long tracks in the cells known as microtubules, which if you like are the railroad tracks of the cell, along which traffic can be directed, that it's from these that they grow. And so they're constantly growing from this little point here. And so if you label with one of the growing proteins, a marker for the growing, then that's in green. And you'll see that it's focused very tightly here because there's constantly new bits being added. But you'll be able to see these little tracks here. And then you can see our little secretory lysosomes or granules with all that loaded with their toxins are lined along the tracks, and they move along the tracks, and they like best to move towards this structure here, the centrosome. So if you move the centrosome up to the target cell here, you'll see that you'll also move all of these little poisonous sacs. So here they go, and you can see that they all cluster very quickly, and they're delivered right to the point that's been identified by this clever structure, the centrosome, which is scanned and said, this is the point to go for. And this was done by Yukako Salna, a Japanese postdoc in my lab. So what does it look like if I can show you a movie that shows you more or less the whole thing? So this is one of my favorite movies. They're hard to capture the perfect series. But here is our killer cell, and we've labeled the outside of it with in green, and then in red we've got the secretory granules with all their cytotoxic proteins. In blue is the centrosome, but it's a bit difficult to see, so we put a little golf ball there. What you'll see is as soon as it comes in and recognizes a cancerous target, it puts this meshwork, which is known as actin, across the interface, but then it sort of clears it away, and somehow it coordinates the clearing away with the movement of this centrosome, which brings forward the poisonous secretory granules in a very tightly sealed area and a very precise delivery. So you're not going to kill the cell next door. You're just going to kill the target that's there, that's been recognized. So it's an exquisite way of delivering very, very accurately from one cell to another. The centrosome is really dynamic. It can scan, it can move quickly, and it's really it really is a very effective mechanism for directing killing and making sure it's accurate. So what happens when those little poisonous granules have been released? So I've told you that this is sort of, if you like, a meshwork which is called actin, and it's around the whole edge of the cell. If you like, it's like a skeleton that gives the cell its shape and then lets it change shape very quickly. But look at what happens once these little granules have been secreted. So here are two granules that have been secreted. In this case, we've had to use a different kind of microscopy because it all happens really, really fast. And so it's as though you are the target right now, and this is the killer cell, and this is what you're seeing being delivered towards you. So here is the actin, which is what sits where the cell has clamped on, and here are the little granules. So you'll see they burst open. Then look what happens. That meshwork comes over immediately and turns it off. So you deliver the hit and then you immediately turn it off. Well, that makes a lot of sense because you could do a lot of damage to the cells if there was an excess of killing. So that tells you how they can turn it on and turn it off. But what happens once that T cell now, it's gone along and it has taken aim and it's killed. So here's its target. Here it goes in the centrosome saying, okay, this is the spot. We haven't imaged the granules, but they're being delivered. The killing's done. This meshwork comes over and says, okay, you're done now. What do you do next? Well, I told you they're serial killers. So off the cell goes, and it goes, oh, another cancerous target. Off we go. Let's move that, find a place to secrete, and kill again. All happening within 10 minutes. Absolutely incredible. So a single cell can kill multiple targets, one after another. Sorry. Yeah. So nevertheless, once I've shown you how good they are at killing, but how can these killer cells control how much they kill? And here's another spectacular movie with a wonderful microscope that was made in the USA, Genelia Farms, by somebody who won a Nobel Prize for microscopy. And you can see the killer cell. You can see how angry it appears as it latches on and kills, in this case again, a cancerous target. So how does it control that? Well, again, what we've done, it's using a different microscope. We're showing this skeletal actin area that surrounds the edge of the cell and allows the cell to change shape. And this shows you, this is a cell and it's moving along this direction, and this is looking down. So you've got another camera looking down at the same thing. If I get that moving, you can see that the way these cells move is they put out little protrusions. And then actually at the back, they always have this bit that sticks up. It's a bit like a cat with its tail stuck in the air. I don't really know why, but it does. I'll play it again, just because it's really lovely to watch. And so you can see that they've got little protrusions feeling their way, looking out for the next cell to kill. And what happens, as I've shown you, is that when it meets its target, then you get a real change in the way it organizes its skeleton. And I just want to show you this really high resolution. You've seen it with the lower resolution, but here, what you can do is you can take a single slice with your microscope through just the center of what's going on here. And you can also reconstruct another, which is as though you're the target looking at the killer coming in. And you'll see that it gets rid of the cytoskeleton. It gets rid of, imagine it as a sort of very tightly woven fisherman's net that you can use as curtains to open and close what you're going to allow in and out of the cell. I'll play it one more time. You can see that when it latches on, it opens these curtains. And what happens when it's killed the cell through, this is, so we can only capture this at a slightly lower resolution. So here's again, these fisherman's net curtains. And here is a target. And this time we've put a little dye in the target so we can tell exactly when it's killed. So what you'll see is that first it opens this curtain, the cell is killed, and look, it closes it again, exactly as I showed you. So it's a very, very clever way of saying, okay, we're going to allow some toxic granules out, but now we're going to shut them off again. And so, yes, I can play one more time. It's the minute we see it killed, it shuts it down again. So this is this special barrier known as actin, which controls the amount of killing that any given cell is going to produce. But you've seen by now a lot of different movies of these cells. And you can wonder, well, are there other ways in which these can be controlled? So here's another of these movies, very high resolution, showing it latching on. So there are the other aspects of this that you could control. Well, yes, actually, the way that cells, the killer cells, which are the T cells here, recognize the target is all controlled by very precise little receptors that stick out and recognize something on the target. And these interactions can give either a very strong signal to the T cell, going, whoa, okay, kill, really bad. Or it can say, somewhat bad, kill a bit less. And then how does that translate to everything I've shown you? So Gordon Fraser, who is a student in my lab, has done a really nice study, which we were hoping to get published now, where he's compared what happens when you've got your killer cell here with a strong signal or with a weak signal. And I think you'll immediately see there's all sorts of different things that change. So in red, he's marked these because he was mapping them so he could measure everything. So the granules are going in red and the centrosome in blue. The target cells at this time have their membranes white. But look at the strong signal. Here it comes. So here's the interface marked with white. It does everything I've told you. That the centrosome says this is the point, off you go granules, and it latches on, and it's all over very fast. It probably only needs to deliver 1 or possibly 2 little granules to kill a cell. It saves the rest to go on and kill the others. But now what I want you to see is what happens with the weak signal. So the weak signal, the cell can't quite decide. It's sort of lugubrious, I would say, in T cell terms. And it eventually kind of latches on, but it doesn't really round up, and the centrosome dithers, and eventually it might be killed, but it might not. So you can tone down each different step of this little killer cell to control just how strong a response you can get in the immune system. So I just wanted to come to the end, towards the end with, well, what happens when killing doesn't work? And actually there are some very rare, thankfully, genetic mutations that prevent T cell killing. And these lead to a particular type of what's known as an immunodeficiency. But I just want to illustrate that here this is a normal killer cell releasing its little granules, and you can see that the cell, as the cancer cell's killed, it's blebbing all the time. And here is one of the genetic mutations which stops these granules releasing their contents. And you can see that it latches on to its target. The target actually embraces it, but nothing happens. There's no killing, and actually the T cell gets stuck there. It can't move away again. So there are many other different immunodeficiencies, and those have been enormously helpful in identifying all the different proteins and genes involved in making a killer a good killer cell. But what I've told you is how good these cells are. And in fact, these cells are so good that a few years ago, they revolutionized cancer treatment. And this is from 2016, when there was a headline in the ITV news saying extraordinary treatment could be cancer breakthrough. Well, there's no could be about it. It clearly has been cancer breakthrough. And it's these killer T cells that underpin the successful new cancer therapies. And I just want to show you with a very simple cartoon, just explain to you exactly how this is working. And so there are 3 different types of cancer therapies, but I want to focus on these 2 because these are the most commonly used. There's what's known as a checkpoint blockade. And what that means is that cancer cells put out a don't kill me signal. They're good at that. That's one of the reasons they're so bad. And that means that although you've got good killer cell, they just can't recognize. They're given a signal to go away. So you can give antibodies, which are produced in the lab, which can block the don't eat me, don't kill me signal, don't kill me signal. And so you add these antibodies and they block the don't kill signal. So here they are blocking it. And that allows then the killer cell to go on and kill. So that's checkpoint blockade. You relieve the checkpoint, if you like. The other way of doing it is when, for example, you have a patient and there's a specific cancer, and your killer cells aren't recognizing that cancer very well. You can help them on their way. You can take a few blood cells out of the patient. You can modify them and stick a little receptor that will recognize their cancer in, and you put these cells back inside the patient, and then these modified cells can kill cancer cells. And these are known as CAR T cells. So these two treatments have been amazing, and they really have improved, cured many cancers, melanomas in particular, for many people. The other area in which these treatments have appeared to be very promising, useful, in fact, are during the course of the COVID-19 infection. I'm sure you're all familiar with the fact that while many people can just get the first stage of the disease, they can be ill, but it doesn't progress to anything that is particularly severe symptomatically. But some poor individuals do seem to move on to a severe inflammatory response. And this is almost because the immune system has gone too far and hyperactivated, and you need to turn down this hyperinflammatory phase. It's known as a cytokine storm. And people have found that using an antibody against one of the components of the of this response, so using that checkpoint blockade mechanisms I showed you, that this can be very effective in turning that down and in helping the really bad stages of some COVID-19 infections. So it's become apparent that these immunotherapies are not only good for treating cancer, but actually they're good for treating a much wider range of infectious diseases than anyone thought of at the moment. And at the heart of these treatments are the killer T cells. All you're doing is enabling the killer T cells to work. So one of the things that I did I just want to finish with this last two slides, that one of the things that occupied much of my time during the lockdown, the pandemic, was preparing for a report which I had been asked to write by Imperial College Global Health, together with a panel of experts, which was about realizing the promise of immunotherapy. And they're really a global health institute. And the idea is you've got this fantastic new immunotherapy, which works so well, but it's really restricted to the number of wealthy countries that can use it. And in order for the benefits to be realized, then there are a number of strategies and a number of challenges that need to be addressed in order that it can be rolled out to everybody, regardless of where they are. And so these were the kind of areas that we looked at, And if you want, you can look this up on the Wish. There's a report, it's written so that it should be understandable by everybody, one hopes, because it was written for politicians, policymakers, so it's meant to be persuasive. And it was a fascinating area, and I'd be happy to discuss these if you wish. But I wanted just to finish with acknowledging that none of this would be possible without a fantastic team who are constantly changing a little bit, but here's the current team. If you like the movies, we have 1-minute versions with a 1-minute explanation of what's going on. They're all on our website. And thank you very much. I'd be very happy to take questions.